Hello students! We're going to talk about virus and prokaryotes in this video, and let's get started. First one, what is the definition of a virus? Virus is non-living. That's one important thing for you to remember, not too difficult to remember. A non-living particle made out of proteins, nucleic acid, and sometimes lipid. So it's important for you to remember the different parts of a virus. So we have proteins, nucleic acid, and lipids, right? If you look at this picture under right here, look right here, you have the protein shell of a virus, which is also called capsid, C-A-P-S-I-D. And then there's also the nucleic acid part, which is, it could be DNA or RNA. As you can see on the left, you have a bacteriophage, a bacteria infecting virus that has a DNA genome. And then on the right side, you have an RNA genome of influenza virus that causes flu. So you want to make sure that you understand and remember that every single virus has a protein coat, a capsid, which is also called a capsid, and nucleic acid. And in this case, it could be DNA or RNA. Well, in all the cases. Most viruses can only be seen with an electron microscope, right? That makes sense. Otherwise, you know, if we can't actually see the virus, we wouldn't eat that apple that got dropped on the floor. If you eat apples. I don't. What is the viral genome made of? Okay, like I said before, they can be either DNA or RNA, and the size of the genome ranges from a few genes to hundreds of genes. So obviously we as human beings have a really large genome because we have a lot of things that our DNA needs to code for, but for virus, because they only have some nucleic acids and proteins and maybe sometimes lipids, and carbohydrates, then it really doesn't need that much genome, but it still varies just like all the animals and plants. Our genome sizes vary, right? Now, how do viruses reproduce? It's important for us to understand how viruses reproduce so that we can come up with drugs that can attack the viruses so that we don't get as sick, hopefully. So just a few basic ideas. Viruses can only reproduce by infecting living cells. One reason why we say viruses are not living is because they cannot live on their own and they cannot produce anything on their own. They cannot produce its DNA. It can't do DNA replication. It can't do transcription or translation. They have to do all of the body mechanisms by infecting other cells. Most viruses only infect specific kinds of cells. What that means is that different types of viruses can attack different types of living organisms. For example, the human flu, right? The flu that we get came from some types of flu came from other animals, and those were viruses that used to attack other animals, but then they evolved to be able to attack us as well. And those viruses will not be able to attack plants or bacteria, most likely. So different viruses are very specific for different organisms. You have viruses, and it has a range. One virus, you have a range of different types of hosts that they can live on, and then a different virus will have a different range of hosts. The next word that you need to absolutely remember is called bacteriophage. So this word, you can split it into two parts. It means bacteria. Phage is it means eating. So bacteriophage is a virus that eats bacteria, basically. But it doesn't really eat the bacteria. It's a virus that attacks bacteria, infects bacteria, okay? So those bacteriophage are harmless for human beings. Now, here's a picture showing you the basic steps of a typical bacteriophage, which is a virus. So you want to kind of remember these steps even if you don't see a picture of it on the test, okay? But I'll still ask you about the steps. So the first step is this bacteriophage, or this virus, attaches to the surface of a cell. And this one is a bacteria cell. First off, bacteriophage obviously attacks bacteria, but you can also see that this bacteria has a circular DNA, which also tells you that it's probably a bacteria. So the first step is the attachment. The second step is the entry of the phage DNA and degradation of the host DNA. So what you see is that this bacteria kind of squeezes this DNA out of its body. This virus squeezes out this DNA into the bacteria, and then at the same time the bacterial DNA get chopped up. The next step Well, this capsid shell, this protein shell of the virus will just fall off. It's useless now. It's done its job. Now, this DNA, after it enters the bacteria, oh my God, after it enters the bacteria, it will kind of tricks the bacteria into a state of mind where the bacteria will actually do the job for the virus. So it will make copies of the viral DNA. It will do transcription and translation to make the viral protein capsid, so that many more viruses can be produced. The fourth step is assembly. So the DNA genome and the capsid are produced separately because one is DNA, the other one is protein. So those are produced separately. So we have to reassemble it. We don't. The bacteria cell will allow the virus to reassemble so that you have full viruses. And now that comes to the last part, the really terrible part, 5, the release. So this step is also called lysis. So lysis means cutting up of the cell membrane. So the virus will make the bacteria cut up its own cell membrane so that the viruses can come out of this one cell that's being attacked, and the hundreds and hundreds of viruses can then attack other cells around. And in case you didn't know, bacteria also usually live in colonies, so there'll be plenty of other bacteria to attack. It's going to be a not very fun party. Okay, so all these steps, make sure you remember it. The next one, bacteria. I keep on saying bacteria. So virus actually has 2 different models of reproduction. One is called the lytic cycle. The other one is called the lysogenic cycle. The lytic cycle is the one we just saw earlier, where the virus will inject its DNA or RNA into the bacteria, and then we have transcription, translation, and DNA replication, and then we have the assembly process, and then there's lysis to allow the viruses to come out. However, there are also other types of viruses that can enter kind of a dormant state where it doesn't actively express its own process. It doesn't actually actively make the new viruses. It will insert a piece of DNA as usual, but then this piece of DNA will be inserted and become a part of the original cell's DNA. So the cell cannot tell at this point. The cell doesn't know that this piece of DNA is foreign. And then the cell will do its regular cell division job so that every single daughter cell has a copy of this viral DNA within its genome, unfortunately. And then see, the host cell and viral DNA replicate and divide. And then eventually, when there's certain environmental trigger that tells the virus, okay, this is the time to go. So at this time, the virus will actually kind of allow its own DNA to come apart from the original genome of the cell, and then enters back into the lytic cycle and do the transcription, translation, DNA replication, assembly of the new viruses, and the lysis process to allow the viruses to come out. So in this case, it was the lysogenic cycle will start right here, enters this cycle, this lysogenic cycle, re-enter the lytic cycle, and allow viruses to be reproduced. Scary thing. It's a scary world out there. During the lytic infection, so now here's the verbal explanation of this process. During the lytic infection, the viruses replicate immediately after the infection. During the lysogenic infection, the viruses incorporate its genome in the host genome, right? Like we said, it inserts the genome into the host genome and stays inactive. We call this prophage. You don't have to know that. The prophage becomes active, and viruses are produced when there are triggers from the environment, such as radiation, heat, chemical stress. Now, this is just an interesting thing that you're also required to know about. A retrovirus. A retrovirus is any virus that has an RNA genome, because normally we go from DNA to RNA to protein, and a retrovirus does kind of the opposite. It goes from RNA back to DNA, and then it goes to from DNA, RNA, protein. So this process is very interesting for sure. Now we're going to move on to prokaryotes. There are 2 types of prokaryotes to bacteria, archaea. Bacteria has cell wall, and the bacterial cell wall is called peptidoglycan. As long as you know the bacterial cell wall is different from the fungi cell wall, which is also different from the plant cell wall, that's good enough. And then here's archaea again. Archaea also have cell wall, but their cell wall lack peptidoglycan, and Right now, we don't think any of the archaea are bad for us, but there are a lot of bacteria that are bad for us. Not that it matters to them, you know. Their membranes contain different lipids, and the DNA sequence of archaea is more similar to eukaryotes than it is to the other prokaryote bacteria. So it's important for you to remember, although bacteria and archaea are both prokaryotes, we are actually, well, the archaea is actually more closely related to eukaryotes, such as us, compared to prokaryotes, the other prokaryote, the bacteria. Again, here's a review on how do prokaryotes reproduce. We start with this prokaryotic cell. The DNA will make a copy of itself, and then the DNA, two DNAs will be pulled apart, and the entire cell will be pulled apart, and the membrane will grow. I see this is a stretch out version of the cell, and then eventually splits into two daughter cells. This process is called binary fission. Make sure you know that. You've learned it so many times. I believe in you. Now the last one, is it? I'm not sure if it's the last one. How do prokaryotes allow exchange of genetic information? This is called conjugation. You have a donor cell that has a plasmid that gives it certain features that the other one doesn't have, and then it will form a pilus in between the two cells. This pilus will allow this piece of plasmid to make well, first off, it will make a copy of itself, and then it will send the copy over to another cell through the pilus. So they're sharing genetic information, right? So it's kind of like if humans are able to do that, I can stand next to you, and then I can just give you a piece of my gene. And I don't know if that's a good thing, but it's just very weird. This is what conjugation is. Why are prokaryotes important? We talk about roles of other organisms, right? We learn about it. There are reasons why we learn about it, whether you care too much or not, but I care. Prokaryotes can be decomposers. They make sure that that our planet doesn't look like trash, although we're doing a pretty good job making it look like trash. They are decomposers. They can be decomposers. They can be producers, which means they can produce oxygen for us, beneficial for us, not that they care, but lucky us. And nitrogen fixers, which means they turn the nitrogen into in the air that when we breathe it in, it doesn't work for us, but they can turn it into another form of nitrogen that other living organisms can use. We also use bacteria to produce food and drug and genetic engineering. You know, there's bacteria living in your gut to help you digest. So those are all good things. No, I think this is actually the last one. Okay, this is actually the last one. A word called pathogen. A pathogen is a disease-causing cell, such as bacteria and virus. So anything that's, you know, any tiny thing that attacks your body and is bad for you is called pathogen. So make sure you know that. Bacteria can cause diseases by destroying living cells or by releasing chemicals that upset homeostasis. If you can remember, homeostasis is very important. You want to make sure that, you know, for example, your blood sugar has to be at a certain level. It can't be too high, it can't be too low. Your blood pressure has to be at a certain level. It can't be too high, it can't be too low. Your body temperature, all of that. Bacteria can ruin everything. All right, this is the end of the video. Go on, watch the next one.